calcium. Calcium, atomic symbol Ca, is a white soft metal. It's a divalent cation. It's a highly reactive material. It is one of the premier ones that we find as a divalent cation that is very abundant everywhere. It is readily available in most soils. A lot of soils have bedrock that's made out of calcium or a calcium type material. You've got calcium that is released into the environment. It stays around in the soil profiles. It is readily available in most soils. Calcium, when it gets into the plant, is actively excluded from the cytoplasms. Being a divalent cation means it's highly reactive, but it is quite limited in where it actually occurs in the plant. It's not easily migrated within the plant. Research has shown that strontium may substitute for calcium if calcium is deficient. That's not saying you've got a lot of soils that have a lot of strontium in it, but if calcium is deficient, then you could actually use strontium to substitute for the calcium. Let's look at some of the metabolic functions. We always talk about a cell wall. Calcium is a divalent cation. Its major function is in the middle lamella, which is made out of pectin. Pectin being certain types of sugars often have negative charges to it. Positive charge of the calcium becomes very important. These organic acid parts in the pectin often carry negative charges, and the calcium comes in as a divalent cation and will bind onto both sides of that particular molecule. As it does that, it actually pulls the cell wall in. It holds the cell wall tight. Once that happens, that calcium is no longer available for anything. Its major function is in the middle lamella. This has been shown in quite a few different plants. It is bound within the tissue. It is not mobile. Once it gets in there, it doesn't get out. It doesn't move. Calcium can actually be problematic if it is soluble inside of the plant. It's the pectin in here that really makes the difference, because the pectin has these organic acids in it. The calcium will substitute the hydrogens, pull these together, and bind them. When they make jelly, you often have to check your calcium levels, your acidity levels, to make sure that the pectin will bind properly. If the pectin is too acidic in making jelly, what will happen is you won't get proper binding. Pectin is made up of rhamnogalacturon and homogalacturon domains. Galacturon refers to galactose. So these are certain types of sugars, and they have these organic acid parts in it. The homogalacturon is a linear polymer of 1,4-alpha-linked D-galacturonic acid. You're looking at somewhere between 100 and 200 residues long, which means it's got 100 to 200 molecules of this. It's not quite as large as cellulose, but it's still the same sort of thing. It is a very important material in plants. Deposited in the cell wall with 70 to 80% of the galacturonic acid residues methyl esterified at the C6 position. You're going to have branched materials rather than straight line materials, which allow them to permeate and actually bond things together. Metabolic functions within the cell wall. Hydrogen may substitute calcium during expansion of cell wall and during ripening. This causes slippage of the cell wall. As it causes slippage of the cell wall, it allows things to expand. If you take the calcium out, the cell walls are no longer bound together. They can slip and they can move this way. If you've got cell wall expansion, you've got that. If you've got ripening and the acid gets in there, it substitutes the calcium out. And as that does that, it makes the calcium a little bit more available, but it will also cause the cells to fall apart. If you look at this picture of the cell, the middle lamella is represented by this area in here in the center between the two cells. You've got cellulose being extruded out. You've got the pectins filling in this area in here. As they do that, the calcium comes, it binds, and it holds everything together. Metabolic functions of calcium on membranes. Calcium bridge phosphate and carboxylate groups of phospholipids. When you look at a phospholipid, you see the phosphorus group. You also see the carboxylate group that is on the fatty acids, which is where you're getting your binding onto your glycerol. These negative charges can be bound together by calcium. It can add rigidity to phospholipids. So calcium has an apparent function in membrane rigidity. On enzymes, most importantly, calcium doesn't activate enzymes. Other ions are important as far as enzyme activation. Calcium is not one of them. What calcium does, there are calcium-dependent ATPases that actively pump calcium ions from the cytoplasm into the vacuole. into the endoplasmic reticulum and into the mitochondria. That basically takes the calcium out of the cytoplasm. As you have it pumped out of the cytoplasm, that is good for the plant because, again, the same way it's a divalent cation, it can hold pectin together, it can pull other molecules in where we might not want them to be pulled in. This prevents competition with magnesium. Magnesium is a very important part in there. Remember, magnesium is found in chlorophyll, which means if you've got competition, you're not going to get the magnesium in chlorophyll, and that's not good. The pumping of the calcium out of the cytoplasm prevents inhibiting cytoplasmic or chloroplastic enzymes such as PEP carboxylase from functioning. If you've got something like a C4 plant, PEP carboxylase is extremely important. If you've got calcium in the middle of it, it's going to slow that down. Calcium has to be removed out of the way so it's no longer in the cytoplasm. It also prevents calcium from precipitating organic phosphate. Having calcium out of the cell is very important for the cell to function properly because calcium binds onto things very effectively, and as it does that, it can cause more problems than we would really want to have with it. Other functions with enzyme. There is a calcium binding protein called calmodulin, which will hold up to 4 calcium ions. It may reversibly bind each molecule of calmodulin. The calcium comes in, it is bound onto this. This allows calcium to be transported around or moved from point A to point B. It becomes very important. If it can pick up the calcium in here, and the calcium's in the middle lamella, it can actually cause a softening of the middle lamella area. This particular protein becomes very important. The binding exposes 2 hydrophobic areas on the protein that enables the protein to bind to hydrophobic regions on a large number of key enzymes and then activate them. So the calmodulin is a very important type material on plants. Calmodulin is also important in animal systems. Calcium is important in the structural and functional parts of animal systems as well. This is the exact same protein that's found in both plants and animals. So it's another example of where you can find similarity between these materials. Metabolic functions, interactions of calcium with phytohormones. There's question as to whether or not this may not be the same sort of thing as the interference with enzymes. Apparently, there are some reports that you get interaction with phytohormones. Auxin-mediated growth is due to protons, which substitute the calcium. When you've got auxin-mediated growth, and this is going to cause, things like epinasty and bending towards the light, you have to have the protons come in. They relieve the calcium of binding the middle lamella. This is going to allow the cell walls between the 2 cells to slip, which will allow bending of things in a certain direction. When you look at auxin functioning, it directly affects calcium. You've got an important part in here as well. Pollen tube growth may also be mediated by calmodulin. When the pollen lands on the stigma and it starts creating that pollen tube that goes down the center of the style and finds the spot in the ovary where it can get in, this is very important. In order to have that happen, you have to have the proper removal of calcium, which the calmodulin can do. Then the calmodulin can release the calcium back in after the pollen tube has transferred its genetic material on.